There are a number of different ways that mutants, mutagens, and carcinogens are identified. And before we start talking about some of these different techniques, it's important to understand the difference between what we call a wild type and a mutant. The wild type is really just the normal version of an organism. So by normal, this would be without mutations, without changes happening, and the mutant is going to be the one with mutations. And so that's the one that we would expect to be a little bit different from what we typically have. So keeping those words in mind, if we talk about some of the different ways that we identify mutants, positive selection is going to be a process that involves selecting a mutant by eliminating, killing off all of the wild type phenotypes. The example that we have right here is you have a bunch of cells that are being grown in a particular media, just a normal media. And then we're going to take those cells and we're going to plate them on media that contains an antibiotic, and we have media that does not contain an antibiotic. So typically this particular strain, this wild type, is not resistant to antibiotic. So when we plate it on the plate that doesn't have antibiotic, we get lots of growth. And we have no way of identifying whether there's any mutants there. But if we take that same population and we put them onto a petri dish that has some antibiotic in it, now we might have just 1 or 2 cells that actually grow into colony forming units. So here we can specifically identify that that cell right there did actually get mutated because now it's a little bit different, and in this case it's able to resist or not be killed off by this particular antibiotic treatment. That's the positive selection. We can also do negative selection. Negative selection is a little bit more work. This is going to be selecting a mutant by comparing its growth really on 2 different medias. So in this case, we're looking for something that grew on one and now it doesn't grow on the other one, even though the wild type still grows on the other one. So in other words, the wild type grows on both media, this only grows on one. So what you can see in this image is that we've got some kind of normal standard media. We've got a bunch of cells that have been grown on that. And then we take a cloth or some type of surface and we're going to stamp it on top of these cells so that we basically gather some of the cells. And then we stamp those onto a fresh petri dish that has a different type of media. That will result in wild type organisms growing, but if there are some mutant ones, then they're not going to grow. So here you can see down at the bottom that we've got kind of a missing colony there. So it was there in the original plate, now it's missing. That we know is going to be a mutant cell. So we would have to go back to the original plate, and now we can collect those cells and grow them more so that we can study them further. The Ames test is going to be a useful test for identifying possible carcinogens. So this means chemicals that can potentially cause mutations and later cancer. So what we're looking at here is we've got some bacteria that typically would not be able to grow on this media. So we take those bacteria and we add a suspected carcinogen, a suspected mutagen, and basically we look at whether or not it is able to restore growth to certain cells. And if we end up getting cells that are now able to grow on this media, they would be assumed to be mutants, which means that yes, this is a carcinogen because it is able to cause mutations. And I know we're looking at bacteria here, but if something is able to alter the DNA of bacteria, there's a good chance it would also be able to alter the DNA of humans or other animals. Genetic recombination is a process that can create some variability in the DNA sequences. And specifically, this is going to be the exchange of nucleotide sequences between 2 DNA molecules. And we're talking about 2 DNA molecules that are there present inside the same cell, possibly inside the same nucleus if we're talking about a eukaryotic cell. And what you can see is that we start off with 2 different DNA molecules. They actually get a cut in them and they swap pieces with each other. This is something that is actually quite common when we're talking about sexual reproduction. That would apply to eukaryotic organisms. Now, when we do get these new DNA molecules, so they have a new combination than what they had before, we do refer to those new organisms as recombinants. Okay, so recombinants, they are the product of recombination. The DNA is going to be made up of some different pieces than what it was made up of before. If we look at another type of genetic variation, this is going to come from horizontal gene transfer. Horizontal gene transfer is the lateral passing of genes between organisms. Basically, it's not vertical gene transfer. Vertical gene transfer is going to be the passing of genes from parents to offspring, and then to their offspring, and then to their offspring. Horizontal gene transfer, the way that I think about it, is that you have two organisms already, and they're just going to swap some DNA with each other. So there are different ways that this can happen, and it is quite common when we're talking about prokaryotes. One way that this can happen is through the process of transformation. Transformation is going to be when a recipient cell kind of takes up, absorbs some DNA from the environment. So this is just DNA molecules that happen to be outside of that particular cell. So you can see that happening. We've got maybe a cell that died, it ruptured, pieces of its DNA are kind of littered around, and then you've got this other bacterial cell that's able to bring some of that inside. And not only is it able to bring it inside, but it's able to incorporate it and add it to its own chromosome. So it is changing its own DNA by incorporating that foreign DNA. That cell that's able to bring that DNA in and incorporate it, that is what we refer to as a competent cell. So this was a competent cell in this picture. Another way that we can get horizontal gene transfer is with the process of transduction. Transduction is going to be a transfer of DNA, and this is going to occur with the use of a virus. This is specifically a virus that infects bacteria, and we call those viruses bacteriophage or bacteriophage. So in this image, we see that you have a bacteriophage that attaches to a host cell. It injects its DNA, and that DNA is going to get, of the host cell, is getting broken up into bits and pieces, and some of it actually gets inserted into that bacteriophage. Now, when that bacteriophage attacks the next cell, it injects that DNA, and it's very possible that that DNA gets incorporated into the chromosome of the new cell. So it is still, bringing some new DNA in and incorporating that DNA into the chromosome, but the way that the DNA is being kind of gathered and inserted into the new cell is through the use of a bacteriophage. The third type is conjugation. Conjugation is going to be a passing of plasmids between two cells, two bacterial cells. In order to do this, a cell would have to be able to form that specific structure that you see connecting the two, so we call that a conjugation pilus. And so they would need an F plasmid, a fertility plasmid, in order to do that. So that's what we do see in green here. We have a cell that has the fertility plasmid, so it's able to actually connect to a neighboring cell, and once it connects, it's able to make a copy of its own plasmid and send it through that connection to another cell. So when we're talking about conjugation, you have a donor cell, and then you also have a recipient cell. The donor cell has to have that fertility plasmid or the F plasmid. If it doesn't have that F plasmid, it can't make that conjugation pilus and connect to the recipient cell. Now, sometimes that F plasmid information is not on a plasmid, and instead it's actually part of the actual bacterial chromosome. And these would be cells that we call HRF cells. So HRF cells are cells that have taken the information in the F plasmid, they've incorporated them into their genome. That's what we see happening in this image right here. In this image, you have a cell that has taken the F plasmid and incorporated it and made it part of its own chromosome. Because it has that, it's able to make that connection, make that pilus with the recipient cell, and then it's able to actually copy some of that information and pass it to the recipient cell. This picture right here just shows us a comparison of the three main ways that we do have horizontal gene transfer. We've got transformation, which is picking up extra DNA that's just kind of laying around and incorporating it into the chromosome. We have transduction, which is still bringing in foreign DNA and incorporating it, but it's going to be mediated by a bacteriophage or a bacteria virus. And then conjugation's actually passing the DNA from one cell to another. There are certain requirements for cells to be able to do that. They have to have the fertility plasmid, or they have to have taken the plasmid and incorporated the instructions into their genome. One final way of getting some alterations in the DNA that we do want to cover here is with the use of transposons. Transposons are pieces of DNA that are capable of moving from one location to another. And sometimes they're commonly referred to as jumping genes. If you look at the cell right here, it has a DNA molecule, big chromosome, it's purple, and then we've got a transposon, which is a smaller portion of the molecule, and it's able to copy itself and then insert into another location. It is possible for them to jump onto plasmids too. And if we get jumping onto plasmids, the plasmid can move to another cell. That could happen with the conjugation process that we just looked at.